Great. Okay, thank you for inviting me to come speak with you today. I'm going to talk about the ins and outs of genetic testing for epilepsy. I'm a clinical molecular geneticist. I work at a diagnostic laboratory. So this is really from the perspective of someone doing the genetic testing and trying to communicate the results back to the physician and the family as best as possible. And here's some tips for understanding what you might see in a genetic report and how did we even get there. So I wanted to start off with the methods. So what are the methods that we use to do genetic testing in a diagnostic laboratory? And here's sort of some of the common methods we might use if we're trying to interrogate at the gene level. So traditionally, a while back, you would do single gene testing. This is rarely done now, but still sometimes useful if the clinical diagnosis is highly specific to a single gene or a single gene variant. More often we see a panel, which is essentially doing single gene testing for a lot of genes all together at once. This is great for when there's a clinically indicated panel that matches a phenotype. And this allows us to test many genes at one time instead of just one at a time and having to go back and test more and more and more. More commonly, we're seeing an exome or genome. So this is often when a patient has already undergone a diagnostic odyssey and still has no answer. This is great when an individual has large regions of homozygosity, possibly more than one thing maybe contributing to the phenotype. And then again, for someone who has a very complex phenotype, where again, there may be more than one thing going on or even maybe something somewhat novel, and we've already ruled out everything. That's where exomes and genomes are really great. For individuals with epilepsy, we're mainly seeing the use of panels, exomes, and genomes. This is the technology that is fairly common to at least panels and exomes. Genomes are slightly different, but really similar. So how this works is the patient sends in a sample, either blood, sometimes a cheek swab, sometimes it's a spit tube, and the lab will collect the genomic DNA out of that sample. It's then fragmented, so it's like broken into small pieces. And then often what's called a capture method, there's this hybridization step. So what we do is we create these probes that match to the DNA we're interested in sequencing, and we mix it together with the DNA. And we bind the probes and wash away all the DNA that we don't really, care about in that moment. And then we sequence the DNA that was caught by those capture probes. We sequence all that DNA at once. So it could be thousands of genes to 20,000 genes in an exome. And then there's really sophisticated bioinformatics to map the sequence and make sense of it, and do the variant calling and understanding what is all the data that we're collecting here. Increasingly now, as the technology continues to evolve and improve, not only can we sequence a gene, but we can use the amount of sequencing material or amount of sequencing data we get to make inferences about whether there was a deletion or a duplication in the region. So not only just reading the sequence of that gene, we can understand, was there a deletion of the gene or part of the gene or duplication of part of the gene? And how we do this is we not only sequence, but we look for how much information did we get from any region of the genome. So then we map it compared to all the other people that we sequence that same region for using the same methods, and we look for deviations. So what you're seeing here is a pattern of all these individual samples who had this region of the genome sequenced. So let's say this is KCNQ2, and each one of these is an exon for KCNQ2. We see that most of the time, the patients the samples just kind of merge together. But in this purple sample, we see increased signal in this one exon, and it deviates from the normal. So this may indicate that this individual has a duplication of this exon. Conversely, we see that this red individual has less material, less sequencing material at this region, And this may indicate that this individual has a deletion. So it's really just a good screen, but often labs will then confirm this with another method to make sure that, yeah, we are seeing a duplication in purple individual and a deletion in red individual. When we're thinking about exomes and genomes, again, they use very similar technology that I just described. The exome uses those capture probes to pull all the material, whereas the genome's a little different, and I'll explain what that means. So an exome interrogates 1.5% of the genome, but this is the important 1.5% of the genome that we understand. So this is most of the genes that we know may be linked to disease, or genes where we understand that they at least produce a protein, all the exons, the coding exons for those genes. And again, like I said, it's using that capture base to pull the pieces that we know we understand, and that's what's called the exome. And you can see here in this little apple diagram, an exome is this tiny little apple slice, whereas a genome is a whole apple. And what the genome has is not only just the parts that we understand, the genes and exons, all the stuff in between that we don't fully understand yet. So all the material between the exons, which are called introns, all the material between genes, all of that we don't fully understand. That's 6 billion base pairs of information that we still have to learn about. It tends to be lower coverage because you're covering so much of the genome that just to be able to store the data and understand and analyze the data, you tend to have a little bit less coverage, which just means it's not quite as sensitive of finding certain types of variants. But again, because you're sequencing so much more that we don't really understand yet, this is mostly done on a research basis, but it's increasingly being done in the clinical lab. So Rady's NICU program uses a genome. Often clinically, we kind of have to throw out the regions we don't know, but we have it there. And so we can always go back and if we learn something new about a region, it's there. But often this is still done in the research setting, obviously, so that we can learn more about those parts of the genome that we just don't understand yet. I wanted to also talk about like the future diagnostics of epilepsy and where I think we're going to go past the genome. There's polygenic risk scores or modifier genes. So this is the idea that it's not just one change that causes the disease, but maybe it's the additive effect of multiple changes or patterns of changes that maybe cause disease or increase risk for a disease. Then there's more omics. We talk about genomics, understanding the whole genome, but there's also a transcriptome. So we've all learned about mRNA and with the advances in vaccines using mRNA. So a transcriptome is looking at all the mRNA in a cell and what's being produced in that cell. The next step is once the mRNA is created in a cell, it goes on to become a protein. So looking at a proteome is looking at all the types of proteins that are described or are created in a cell. And then we talk about a metabolome. So this is the metabolics, your metabolics, and what is produced and what is broken down. And I think there's just still so much for us to learn in these different types of omics that are just going to continue to emerge over the next few years. So I want to spend a little bit of time about variant interpretation and reporting. I imagine most people here have received a genetic report. And how are we interpreting a variant? How are we reporting it? And how come sometimes what we say changes? Like we say one thing one day and then a year later, we might be saying something different about that variant and trying to explain how this happens. So a while back, labs were using different language and the ACMG and AMP groups came together to try to standardize the language that diagnostic labs were using so that one result from one lab and a result from another lab could kind of make sense to each other instead of everyone speaking a different language. And they agreed on these terms that I saw Al describe earlier, so I'm going to kind of gloss over them. but we all agree to use the terms pathogenic and likely pathogenic when we're describing a variant that we think causes disease, or we're really pretty certain that they cause disease. The counter of that would be benign or likely benign. This is when we are pretty sure we know the variant does not cause disease. And that likely in between the words is just how certain, like we're pretty certain or we're very certain of a variant causing disease or not. What's in between are variants of uncertain significance, or VUS. And these are variants where they either cause disease or not. In a very simplistic term, we just don't know yet. We don't have enough evidence to say, yeah, we're pretty certain or fully certain that it either does or doesn't cause disease. And we use lots of pieces of evidence in the lab to try to figure this out. We don't want VUSs. We want every variant to fall into one of these other categories so that we can give a clear result. But we sometimes just don't have enough evidence to say one way or the other. Some of the evidence we use is population data. So these big databases like Nomad, our own lab databases, ClinVar. Are we seeing this variant in control individuals or not? What type of variant is it? Is it a missense? Is it just a change of one amino acid to another? Or is it a nonsense where we expect the protein isn't ever even made? And for that gene, what kind of variants do we know cause disease in that gene? We look at clinical findings of the individual who's being tested, clinical findings of individuals who have been reported in the literature, clinical findings of other family members who maybe carry the same variant or don't carry the same variant, but have the same disease. All of that information can help us understand if we're going one way towards pathogenic or the other way towards benign. And then molecular studies. So Al showed all those patch clamp. That's information that we use in classifying the variant. And then we even have these like in silico predicted models that we'll sometimes use as well. All of this information we're weighing and balancing to figure out, does a variant cause disease or not? So here's some examples of what a report could look like. This is an example of a positive report. We'll often say that it's positive, meaning we believe this to be diagnostic. We found the cause of the disease in the individual we were testing. We may state if there's increased risk to biological family members and if maybe others should consider also being tested. And this is, general information, the gene, the variant, zygosity. Was the variant heterozygous? We found one copy of it. Or was it homozygous? Did we find both copies of that variant in that individual? And then the classification, pathogenic, or in this case, this individual also had these other variants of uncertain significance. This is the opposite report. So this is a negative report. We'll often say that we did not find the diagnostic cause for that individual's disease. This does not rule out a genetic etiology. It just means we didn't find it using that specific test. Often further testing may be warranted. We may have to go to a larger test. Maybe we didn't test the right gene. Maybe we didn't test the right region of the gene. There's lots of different reasons why we may not have found something that truly does exist in the individual's DNA. It's also important to note that just because it's negative doesn't mean we didn't find variants, but often we don't report all those benign and likely benign variants. So there's a variance that we find in individuals, but we don't think they cause disease, and often individuals will have thousands of these variants. So it would make the report really complicated to report all these variants. But usually if you're interested or your physician's interested, they can contact the lab and we can provide a list of these variants. They're definitely for you to know, but for simplicity's sake, they're just not put in the reports. Lastly, here's the uncertain results finding. So this is where we found variants, but again, we couldn't decide if we didn't have enough evidence to decide if they're pathogenic or benign. So we'll list the variants, we'll provide that same information, but often in these reports, we'll talk about if there's things we can do to resolve this variant. So often, is there familial testing? Like, can we test the parents? And if we found that, the parents didn't have this variant, that might be enough evidence for us to move into the direction of pathogenic. Or if we found the variant in a parent that did not have the same disease or had no disease at all, that might help us move the variant to benign. So there's usually information in the report asking if there's more we can do to resolve a variant. A lot of labs will also provide patient guides to understanding the report. So I know our laboratory has them for positive, negative, and VUS results, where it'll explain, what does this mean? How do I create a plan with my health provider? What does this mean for family members? And these are valuable tools that you can often find in different laboratories, websites, and your provider can also help you find material that can help you understand the report as well. So I think, though, I wanted to spend a little bit more time on the VUS, because I think that this is like the bane of our existence. There's a variant there and we don't know what it does, but there's things that we can do and work together to help us figure these out. So like I said, one of the easiest things is to test the parents. Was it de novo? Do the parents not have the variant and only the affected child does? For other genes where it's recessive, where you need two damaged copies of that gene, one from mom and one from dad, to cause disease, did we find one of the variants in mom and one in the dad? That shows that, yeah, that child has two damaged copies, no good copy, which likely links to that disease. Other times, family testing, does the variant segregate with other affected family members? And we can sometimes look through multiple generations and see that this variant is segregating with everyone who has the disease, and everyone who doesn't have the disease does not have the variant. And that's really important information for us. Sometimes just doing more of a clinical assessment of the patient. Does that gene and variant match the patient's phenotype? Sometimes the physician can look at the gene and look at the patient and say, there's no way this is a match. Like, this patient, to have this disease, would have to have all these features that your child does not have. So I really don't think it's a good match. And that's really important information that can be provided back to the lab and can sometimes help us resolve that variant. Sometimes further phenotyping might be needed. We may find a gene variant and it's like, I didn't look at X, Y, and Z, but if I had, I would have seen that the patient does have these, and this gene actually might be a good fit that I didn't realize because I hadn't looked for these other individuals. 105 of them, so about half of them, sent family members in for further testing. And we found that when this happened, 30% of the VUSs were reclassified from having done the parental testing. So the vast majority of the variants went to pathogenic or likely pathogenic, and a couple of them went down to likely benign. And the majority of this was because the variant was found to be de novo in the affected child. So mom and dad had normal copies of KCNQ2, and the version of KCNQ2 with the pathogenic variant was only in the child who had the symptoms of the disease. So again, this is a really valuable thing to do when the lab is asking to do family testing. If you're able to, it's really important to, because it could really help resolve these variants. I wanted to talk a little bit about reasons why variant interpretation can change, because I know that can be really frustrating to be told one thing and a couple of years later told, oh, that wasn't right, and now it's something else. I think Al already talked about ClinVar and ClinGen and this effort to share information. And I just highlighting this, that many labs do share information and we learn from each other. So this is a place where information that one lab had that the other didn't have is shared. And now we all have gained information, which can help better resolve variants. I think it's important to remember that the cost of sequencing DNA just continues to drop dramatically. And so this was around the time of the first sequencing of the human genome, and it should be dropping like this, but it's dropping like this. So it just means that we're able to sequence a lot more information and a lot more people. And what that means is we now know more than we knew yesterday, and tomorrow we'll know more than we knew today, just because we have so much more information. So one bit of information, and I think what has led to a lot of variants being reclassified as benign or likely benign, is this evolution of the large databases. So these are like the big databases of control, seemingly unaffected individuals who have contributed their genomic data for the public to see and compare. Back in 2010, we saw the 1,000 genomes. So it was exactly what it sounds like, 1,000 genomes. And at the time, this was fantastic. We finally had 1,000 people to compare information to. But we learned very quickly when in 2014, the Broad Institute released 60,000 exomes, just how much more information there was that we didn't realize we didn't have and we needed. And a lot of variants that we thought were novel in one individual, because it was absent in this original 1,000 people, we're now seeing in the 60,000 people, and it doesn't feel so novel. And then in 2017, the Broad Institute released even more data. We now had over 100,000 exomes and 15,000 genomes, and then even more genomes, 70,000 genomes, just so much more information. So again, a variant that may have looked novel 10 years ago, we now see in other individuals in these large databases, and that provides some evidence that variant might not be associated with disease. On the flip side, as we've been able to sequence more and more data for more and more individuals, we've also learned about so many more genes. And this is a common image that we like to throw up on our images from Ingo Helbig, just showing this trajectory in epilepsy, and this just continues to grow. There's like a new gene every other day associated with disease and often associated with neurological conditions and epilepsy. So we continue to learn more about the exome and the genome that we didn't know. And this changes how we feel about variants that maybe looked like not important, and now they're in a disease-causing gene we just learned about, and now we think this might be pathogenic. I just wanted to spend one moment on sponsored testing programs. I talk about this because 10 years ago, 5 years ago, it could be quite hard to get genetic testing. It is still hard to get genetic testing for some indications, but for epilepsy, we've made this very, very easy to get genetic testing. We've tried to reduce all the barriers that, that could be put in the way, whether it's cost or access. So our laboratory works with biopharma partners who will sponsor the testing. We've got all these sponsored programs in so many different indications. Many of you are probably familiar with the Behind the Seizures program. So through this Behind the Seizures program, which is sponsored testing for epilepsy, it is written really broad. So just one unprovoked seizure. Currently, it's in any child who's had an unprovoked seizure under the age of 8. We hope to expand this to older kids, and we're actively working on that. To date, we've issued over 16,000 tests for individuals with epilepsy. We have 10 biopharma partners who all work together to sponsor this. And the panel right now is looking at 290 genes. KCNQ2 has been on the panel since day one, but as we're learning about more genes, we keep adding other genes into this panel. And this is just showing different quarters and how many individuals were tested in this program. And we originally, it was 2 to 5 year olds. It got expanded to anyone under 5, and then expanded to anyone under 8. And we continue to offer this testing to many individuals, which is, I think, been really great for this field. Thank you.
Thank you very much, Dr. McKnight. I really appreciate that, and a great follow-up on some of the presentations that we had yesterday from some of our other experts. And that brought up the question from some of the people listening about your testing for mosaicism, as that is a very hot issue among our families, and whether some of the variants are actually de novo, or whether they may be inherited from a mosaic parent and may not be showing up. And also the genetic background, because what we see among many of the children with the same variant have very different presentations, and whether that's related to genetic load or the genetic background, and what Invitae and your testing might be able to pick up in both those settings.
Yeah, I'll start with mosaicism. So I fully agree. I think mosaicism is far more common than we ever thought it was. And as the technology has improved, we've been able to see this. We've been able to see that, yes, in instances where we thought it was de novo in the child, the parent was actually low-level mosaic. That mosaicism is challenging because it depends on what sample you're testing. Unfortunately, for most of these genes that cause epilepsy, the level of mosaicism is usually the lowest in blood, which is one of the most common samples that are submitted to laboratories. But I think as the technology continues to improve and become more sensitive, we'll find more and more of these cases. But this is definitely a place where we're still learning. We do report out mosaic variants at our laboratory, and most laboratories will report out mosaic variants. Whether it's how sensitive it is can always depend on the sample and many other extenuating circumstances, but if a variant is seen, it's often reported. The second question about genetic background, yes, this is exactly where I think we still have a lot to learn. Why do two people with the same variant have very different disease? And this is going to be the rest of the information in the genome, or in that we still need to learn. I think that it could be the background variants in both KCNQ2 and maybe other genes that make proteins that also interact with KCNQ2 that may be causing this, but there's still a lot of research to be done. Through the sponsored testing program, we're sharing a lot of this data with researchers or biopharma partners who are all really interested in this and are doing this exact kind of research to try to figure these questions out as fast as possible.